Welcome to Lecture 7, where we will go over the diversity and characteristics of fungi. This diagram represents the current phylogenetic hypotheses representing the relationship between the major clades of extant or living fungal groups. Note that we have an common ancestral protus, which was considered to be flagellated. This then led to the two basal lineages, the cryptomycetes and microsporidians. We then have the chirtids, zoopagomycetes, mucoromycetes, ascomycetes, and basidiomycetes. Current genomic analysis indicates that there are two basal lineages of fungi, the cryptomycetes and microsporidians. Both are considered to be unicellular. Cryptomycetes have flagellated spores, remnants of their ancestral species. Though many cryptomycetes are considered parasitic, they can also be found free-living in soil, marine, and freshwater environments. They can also be found in aerobic and anaerobic conditions. Microsporidians are unicellular parasites of both protists as well as animals. They can produce spores with a unique harpoon-like organelle that aids for their entry into their host cell. Many microsporidians have reduced mitochondria as well as reduced genomes. Similar to most fungi, both of these groups can make a cell wall based out of chitin. The chirtids can be found in almost all environments as well as hydrothermal vents. They are considered to be important decomposers, but they can also be parasites of many different organisms as well as mutualists. A characteristic of this phylum is the production of flagellated spores called zoospores. These organisms can be in the solitary form, or they may also form colonies with hyphae. There are currently approximately 1,000 known species. Members of the Zoopagomycota phylum are parasitic, or they can also live as commensal symbionts on animals. Many of these species form filamentous hyphae. Some parasitic species can cause distinct behavioral shifts in their host organism. Many have sexual and asexual life cycles. In the asexual life cycles, they produce non-flagellated spores. It is believed that the loss of flagella corresponds to the transition of fungi onto land. In the sexual reproductive cycle, they can produce a resistant stage called the zygosporangium that surrounds and protects the zygote. The Mucoromycota phylum is the phylum that is most closely associated with plants. They can be considered parasites, pathogens, mutualists, and some decomposers. The Mucoromycota species are very commonly found on rotting bread, fruits, and vegetables. These species can form zoonocytic hyphae. They can also reproduce asexually by producing flagellated zoospores. Some hyphae can actually aim towards the light to ensure that spores disperse into new areas. However, if a food source becomes depleted, some can use sexual reproduction to also form a zygosporangium. This can undergo meiosis when conditions return favorable. This diagram represents a typical life cycle of a mucoromycete. Note the haploid state in blue, the diploid state in pink, and the heterokaryotic stage in purple. Note that when resources are plentiful, asexual reproduction occurs. This means that the mycelium or hyphae will produce sporangia that produces spores that germinate in the vicinity. However, if food becomes scarce, the sexual reproductive life cycle may take over. In this instance, the mycelia would form different mating types, positive and negative. If these different mating types or mating types from different organisms that are genetically different encounter each other, they may form gametogania with haploid nuclei. This may undergo plasmogamy to form the heterokaryotic stage. This would then be called the zygosporangium. The zygosporangium would then undergo karyogamy to form a diploid nuclei. This diploid nuclei would undergo meiosis to then form the sporangium, which would also form spores. The Ascomycota phylum is also a very diverse group that can be found in many different environments, marine, freshwater, as well as terrestrial. Many of these species, up to 25%, can form symbiotic relationships with algae, cyanobacterium, lichens, or plants. Some are considered to be mycorrhizal with close association with the root system. However, some can live within plant cells and also release toxins to help deter insect predators. Many other species are known as decomposers as well as pathogens. The Ascomycota are called sac fungi. This is due to the presence of sac-like asci that end up producing ascospores. Ascomycots can undergo sexual and asexual reproduction. Many species can reproduce sexually by developing fruiting bodies called the ascocarps. These are the sac-like asci that end up producing ascospores. However, in sexual reproduction, specialized hyphae called conidiophores will produce asexual spores called conidia. In some species, these spores can germinate asexually, or they can undergo sexual reproduction and fuse with the opposite type mycelia. This extends the dikaryotic stage and allows for increased genetic diversity. This diagram represents a typical life cycle of an Ascomycot, in this case Neurospora. In the asexual reproductive cycle, specialized hyphae called mycelia can form a specialized conidiophore. This conidiophore can produce haploid spores called conidia. These will germinate in the surrounding region and form more mycelia. However, sexual reproduction can also occur. In this instance, the mycelium will produce special mating types. If a conidia of an opposite mating type, such as a positive and negative, encounter each other, they may undergo fusion. This would be termed plasmogamy. If that process occurs, a dikaryotic hyphae would result. This would then lead to the production of asci, or singular form would be ascus. The ascus can undergo karyogamy, where a diploid nucleus is then formed from the fusion of the two different mating types. This would produce a diploid zygote. This zygote can undergo meiosis to produce 4 haploid nuclei. These haploid nuclei would then develop into ascospores. These ascospores would further develop and form the asci. These asci contain the ascospores, which are considered haploid. The asci are contained in a structure called the ascocarp. When ready to release the spores, the ascocarp will contract and forcibly eject out the ascospores to the surrounding environment. These spores will then travel to surrounding areas to undergo germination. The basidiomycots include mushrooms, puffballs, and shell fungi. Some can be mycorrhizal, some are parasitic, while others are important decomposers. These species are named due to the presence of a club-like basidium that forms during the process of karyogamy. Similar to ascomycetes, basidiomycots will have a long dikaryotic stage. This allows for an increase in genetic diversity through genetic recombination or fusion of nuclei. The mycelium of basidiomycots can form extensive fruiting bodies called basidiocarps. These basidiocarps may have gill-like structures that are lined with basidia, which are dikaryotic cells. Each basidium can undergo karyogamy to produce a diploid nucleus. As seen in the previous life cycle, this diploid nucleus can undergo meiosis to produce 4 haploid basidiospores. This diagram represents the typical life cycle of a mushroom. Within the mushroom, there are specialized mycelia that will undergo reproduction. In sexual reproduction, 2 different mating types may be produced, positive and negative. If different mating types encounter each other, they may undergo plasmogamy to form a dikaryotic mycelium. This could then lead to the formation of a basidiocarp, as seen in the diagram. This basidiocarp has gill-like structures on the underside of the head of the mushroom with basidia. This basidia can undergo karyogamy, which means that the haploid nuclei will fuse together to form diploid nuclei. This diploid nuclei can undergo meiosis. If this occurs, this can form a basidium that each contains 4 haploid nuclei. Take a look at the inset picture below for a close-up examination of the structure of the basidium with the basidiospores. These basidiospores, once again, are considered haploid, and they can then be released and undergo dispersal and subsequent germination to then form their own mycelia. Some basidium mycots are known to be responsible for what is termed fairy rings. It is believed that these fairy rings were magical because they were rings of mushrooms that would appear overnight. However, we now know that some of these rings have extensive mycelium networks that extend underneath the ground and can be thousands of years old. They only form the ring above ground when they're ready to reproduce.